tissue that we want to know about at first the general specification of the muscle tissue. It means that how we can identify the muscle tissue from other tissues. They have the cells of the muscles have some specific characterization that we can identify them from, for example, the epithelial tissue or other tissue. There are in the human body three types of the muscle, as skeletal muscle, cardiac, and smooth muscle. Why we name the skeletal muscle? Most of these muscles attach to the bones, the skeletal system, because of it in the macroanatomy or in the anatomy, we name them as skeletal muscle. But under microscope or in the histology, we divided the muscle to two types, the striated, these two, and smooth. The skeletal muscle and cardiac muscle microscopically are very similar together, just have some differences. Because of it, at first I explained about the skeletal muscle, then we now just the differences between the cardiac muscle and the skeletal muscle. Thus, under microscope, the skeletal muscle and cardiac muscle, both of these muscles are striated. And another type is the smooth muscle that don't have any striation under microscope. Skeletal muscle cells mostly attached to the bones, but there are some exceptions in them, such as some muscles that are in our face, named as expression muscle, that mostly they attach to the fascia, to the superficial fascia. Because of it, we can show our different senses by the contraction or relaxation of the esophagus. Esophagus wall is the skeletal muscle tissue, but don't have any attachment to the bones. There are some of them, or for example, the abdominal wall muscle are here. Also, they some parts attached to the fascia. Cardiac muscle is just in the heart. Cardia is a word that in the clinic use equally to the heart, such as cardiomyopathy, means that the disease that are for the muscle of the cardia, or cardiac infarction of the heart. A smooth muscle tissue occupies the wall of the hollow organs. All of the organs that are in the internal part of our body, they are occupied by the smooth muscle tissue. Under microscope, as you see here, the skeletal muscle and cardiac muscle have some dark and light line that named as striation. Under microscope, these are because of the arrangement of the proteins in the cytoplasm that we now name later. But the smooth muscle don't have any striation. Here you can see the practical slides in the skeletal muscle, striation, lots of nucleus, the cardiac muscle with branching cells, and the smooth muscle with spindle shape cells. The muscle tissue have different function. The main of them is movement of our body, skeletal muscle, movement the internal organ, the peristaltism, contraction for movement of the food, feces, fluids, or other one that squeeze the fluid or other substance in our body. During the stabilization, they with the bones and joint maintain our posture, our shape in the body. heat generation, by the movement, they can involve in the regulation of the body temperature. The respiratory system muscles that are in the thoracic cavity wall here, or the diaphragm muscle, are some type of the skeletal muscle that involve in the breathing. Support soft tissue such as abdominal wall. Yes, in the abdomen, anterior abdominal wall, we don't have any bone. Yes, this part have just the muscles that support the GI system organs or the reproductive system. In some part, they can act as a guard in the entrance or exit part of some organs, such as esophagus. In the upper part, lower part, have some muscle formed some circular structures as these, they around the organ and form the sphincter. This sphincter act as a guard. They control the entry or existence anything from the organs. And finally, because in the cytoplasm there are lots of glycogen, the muscle tissue can reserve the glycogen in the cytoplasm, can act as a store for storage of the nutrient. At first, we want to know the skeletal muscle. But before starting of this part, the muscle cells are very long compared to other tissues, compared to epithelial cells or connective tissue cells. In the embryonic period, when they derive from the myeloblast cells, they a little are longer than other tissues. Because of it, the skeletal muscle cells, mostly named as fibers, they are equal because they are long. Mostly used beside of the cells being fiber in the skeletal muscle. You know the cell, all of the cells have a membrane, cytoplasm and nucleus. In the skeletal muscle, just their name changed. The cell membrane of the muscle tissue named as sarcolemma. The sarco or myo or are equal for the muscle in the medicine. In the medicine terminology, you know, in the medical science, there are a lot of words that just specific in the medicine, not other majors. That I think you have 2 or 3 credits that know about it. That you use these words in the clinic or in the future that you go to hospital or clinic or others. Sarco and myo is 2 words that use clinically equal to the heart, to the muscle. Sarcolemma means the membrane of the muscle. Sarcoplasm is a plasma or cytoplasm of the muscle cell. That this cytoplasm have 2 main specific content as glycogen that store in the muscle cells, and they can use it for the ATP synthesis. And another one is a red color protein that named myoglobin. That also equal of these we have in the blood as hemoglobin. Hemoglobin is a protein that have red color. Because of it, the blood and muscle, both of them freshly have red color. In the muscle named the myo, that as same as to the hemoglobin can bind to the blood gases and carry out them. The smooth endoplasmic reticulum, named as sarcoplasmic reticulum, and have a special function in the muscle cell. calcium ion. The calcium ion is very important for contraction, for the releasing of the neurotransmitter also from the terminal part of the axon. Because the cells should use low ATP and have easy access to the calcium ion. This ion is stored in the endoplasmic reticulum, smooth endoplasmic reticulum. If you memorize in the cell first part, I explained for you that the SER cisterna have tubular structures. They have a narrow movement that, as you see here schematically, form a network of tubules inside of the cytoplasm around the smallest structures of the muscle tissue that we name them sarcoma, that form a network of tubules that the cells have easy access to them. The other things, the other organelles also are in the muscle cells. Skeletal muscle cells. Skeletal muscle fiber, because they are very long. The muscle cells are longer than other tissue cells. But again, the skeletal muscle cells are more longer compared to the cardiac muscle cells and smooth muscle cells. Why? In the embryonic period, around the fourth to fifth weeks, that muscle want to differentiate it, following the expression of the genes, the mesenchyme cells that explained yesterday about it, that derive from mesodermal layer, differentiate the precursor cells of muscle cells that named as myoblast. Myomuscle blast, the cells that are active. They are immature and active in protein synthesis. They derive from the mesodermal layer of the embryo. The myoblasts in the skeletal muscle fuse together. Lots of them, around maybe 100 to 150 of them fuse together, and they form a very long fiber with lots of nucleus. Thus, we tell that the skeletal muscle cells are long, cylindrical, multinucleated cells because of the fusion of the myoblast in the skeletal muscle. They differentiate to very long cell or very long fiber that, because differentiate from lots of cells, it have lots of nucleus. That finally, by differentiation, they form a long cylindrical cells with lots of nucleus. The nucleus mostly are flat and located under the sarcolemma. Some of the myoblast cells remain as undifferentiated cells. Name them as satellite cells. That these cells are undifferentiated cells. When the muscle tissue damage, they activated by some factors and can differentiate to the muscle cells. They are very long and large, and the unfused cells form the satellite cells that are for repairing. After the fusion of the cells, these cells, the myoblast, synthesis the proteins that in the muscle tissue functionally are two types of maybe millions are of the sarcomere are in the cytoplasm in the cells. In the sarcomere, we can see under microscope the dark and the light line. In the dark line, included the thick filament, included the myosin molecules. That the myosin molecule have a tail and head, and the actin filaments are in the thin filaments. That are the microfilaments in all cells, also in the muscle cells. When we see them under microscope, we can see some the dark and light line. That the dark line included the thick filaments here. That in the physics and also in the physiology, mostly they use as A band, anisotropic. It's related the property of these proteins to the light that they radiated from the microscope to tissues. A band or anisotropic, that included the thick filaments. The I band, isotropic, only included the thin filament. That's, and Z disc pass from its midline. The I band shape between two neighbor sarcomeres. Yes, this part is for this sarcomere, this part is for another one. The old part that included the thin filaments is I band. That just I explain for you because in the physiology explain more for you. We use as more as thick and thin filaments. The thick filaments included the myosin molecules. That each myosin molecules have a heavy chain or tail and light chain or head. That these two parts join together to one part that's named hinge. They act as a hinge, as this one. Hinge means this. That can, the head can open and again close for in the contraction. Around 100 myosin molecules, 100 of the myosin molecules wrapped in their tail and form just one filament, one thick filament, as you see here. They wrap around together by their tail attached to this line. What is this line? M-line or midline. Midline means middle line. In our body also we have it. Yes, separate or divided our body to left and right. In the sarcomere, the M-line divided the sarcomere in two equal parts. In the M-line, there is one structural protein as is, the myomesin, that attach or joint the tail of the myosin to this line. It means these filaments, the thick or thin filament, not free in the sarcomere. They have some places, some sites that attach to them, and some proteins help to them for this attachment, that's named as structural protein. The thick filaments, by the myomesin, attached to M-line, and by another one proteins as titin attached to Z-disc. Another structural protein is the titin. There are around, I see, more than 15 structural proteins in muscle tissue that I just explained, the more important that are in your book. But if you want to more about them, then you can study their cell biology books. Thus, the thick actin general structures are as actin monomers or globular actin. Globular or monomeric. They are as some as small balls. Yes, they are single and displayed in the cytoplasm. When they want to do their function in all of the cells and also in the muscle tissue, want to attend to sarcomeric structures, they should change from monomeric structures to chainy structure. They should join together. As this. The tropomyosin is another structural protein that, what is function? Change the monomeric actin to F-actin or fibrous actin. This protein is a double chain protein that have some site for attachment to actin filament. Change the monomeric actin to chainy actin, such as, for example, as rosemary, yes, that we use for praying. This yes, his name is Rose Percy, or I think your Rose Perry.
Oh.
This is yes. We use for the praying.
Rosemary.
I think
You understand?
Rose Percy, Rose Perry, anything you know it, yes, yes. And included beans, some beans, yes, that they collected by one church, yes. Then in the sarcomer for the thin actin filaments, yes, the G-actin are as same as the beans of it, yes, and one cord like that join them together is the tropomyosin. Thus, the tropomyosin is a structural protein that changes the G-actin or globular actin to F-actin that able to attend in the sarcomeric structure. The other proteins that we see in the thin filament location are the troponin. As you see, have 3 subunits that have some joining site for the different structures that named as troponin. Tn is a troponin. TNA or TNI bind to the actin or inhibit binding to the actin. TNT binds to tropomyosin and TNC binds to calcium ion for contraction. Thus, another one of the structural protein is troponin that joined to the calcium ion and the thin filament can access to it very easily. Thus, in the sarcomer, here in the high magnification, in the midline, there are some proteins that more important of them is myosin, that the tail of the myosin attached to it, the head of the actin as to the side of the sarcomer. They have overlapping to the thin filaments. And in the island or in the thin filament part, if you look at the F-actin, troponin and tropomyosin. In the contraction, just now, the head of the actin myosin attach and detach to the actin filament that we know more about them in the physiology. Explain for you in more detail. T2 bull. What is the T2 bulls? They are some transfers to bull. The cell membrane, yes, of the skeletal muscle. The cell membrane imaginated to the inside of the cell. Because they located transversally and have tubular structures named transverse tubules. These tubules surrounded the myofilaments, and also in some part when they invaginated, they in two sides of them there are the smooth endoplasmic reticulum that they store and release the calcium ion. The T2 bulls are continuous with sarcolemma. Yes, they are continuous to the sarcolemma and with the SER that store the calcium ion involved in the contraction. One T2 bull plus two SER cisterna that are in each side of it form the structures that named as triad, three structures. Yes, one T2 bull, two SER in two sides that are one specific structure of the skeletal muscle tissue, triad, located between the I band and A band. The dark one and light one. Its location is between the I and A band. That is maybe is one of the questions. I forgot that the sarcomere, yes, the different part that I explained for you have just maybe one of the questions, and also the triad or dyad that we know later about them. Neuromuscular junction, what is it? Junction between the nerve and muscle. It's a type of synapse, yes? Chemical synapse. Now, about it in there yesterday. Terminal part of the axon, terminal nerve. Included the neurotransmitters that in the skeletal muscle are the acetylcholine, synaptic vesicles in perisynaptic cells as a nerve release to synaptic cleavage and have receptors on the postsynaptic cells. Postsynaptic cells is a muscle cell. For increasing the number of the receptors of the acetylcholine, the cell membrane have some enfolding, the sarcolemma. That's because these folding are in the neuromuscular junction named as the junctional folds that have receptors for these neurotransmitters that join to them, and finally the ion channels open or closed. Yes, terminal part of the axon, the neurotransmitter release to synaptic cleft, join to its receptors. Yes, and finally the action potential or contraction of the muscle occurs. And finally the additional acetylcholine broken by one enzyme as acetylcholine esterase. Yes. The acetylcholine broken. Some diseases are in this part. Maybe they occupy the receptors of the acetylcholine that finally the person refer in the future, hopefully to you, as a myopathy. They have a weakness in their muscle. Maybe upper limb muscle, lower limb muscle, I don't know, maybe respiratory, the abdominal wall or others. Yes? Two diseases are as the, one of them is the myasthenia and the width. In the myasthenia is an autoimmune disease. You know autoimmune disease?
Yes.
The body produces some antibody against itself. Here, the body produces some autoimmune, some antibodies that occupy these receptors. The acetylcholine released from here. I don't have any receptors for joining itself. In the myasthenia gravis, the bacterial factors occupy these receptors. Thus, this may be treated based on the grade of the disease. Most of the person treated and be safe, but in the myasthenia gravis, mostly the person suffer from it because the autoimmune system disease are with the person in the life. Just they should change themselves. The last topic in the skeletal muscle is the organization of connective tissue. Yesterday we have a same as this in the nerve. Yes? Some connective tissue layers, membrane, enclose the muscle totally and also send some septa to inside of the muscle and divide it to some parts. The epimysium is a type of dense connective tissue. Maybe you saw it previously. You might think maybe you buy it or in your home. Yes, you see that around of the meat there is a very fine white color membrane. Yes, the external part of it. This is the deep fascia in the anatomy or the epimysium in the histology. Epimysium is a layer, is a membrane of dense connective tissue that encloses a muscle that we named it as anatomically, such as the brachialis, biceps, triceps, or the abdominal wall muscles, yes, that have anatomically one name. Enclose all of the muscle. Then send some septa or trabecula to inside of the muscle and divided muscle to small parts, that each of these parts named as a fascicle. These septa divided the muscle to a small compartment and also bring with themselves the vessels and nerves in the deepest part. These membrane, these trabecula named as the perimysium. Perimysium enclose the fascicles, muscular fascicles. What is the muscular fascicle? Are a group of the muscle fibers or muscle cells that each of them sheeted by the endomysium. Come back to previous slides. Epimysium, dense connective tissue that sheeted all of the muscle that included mostly collagen type 1 and connected to the deep fascia. Perimysium surrounded each fascicle. Fascicles are the group of the muscle fiber that contain the vessels. Endomysium is a type of loose connective tissue. Gradually, when they enter to the muscle, yes, the amount of the collagen fibers decrease and form the loose connective tissue that contain the capillary nerve and also the satellite cells that I explained for you previously are in the endomysium. Endomysium surrounded just one cell or one fiber in the muscle cell. Electron microscope just show the endomysium. This is a scanning electron microscope. Schematically, practically, is a special staining for collagen fibers. The peripheral part of the muscle, epimysium, the collagen fibers have red color. Send some septa inside or here and form one fascicle. And in each fascicle are the should see the longitudinal section. Yes, the dark and light lines, cross lines, just see under the longitudinal section, not the transverse section. But this section is good for the finding of these membrane that we see them in the practical class. Any question? Do you have a rest? Yes, around until 9? Then continue for the master. Do you have any question? But here they are located in the center and have a round or oval shape nucleus. The cardiac muscle cells connected together by some structures that named as intercalated disc. Yes, I explained for you there are some similarity between. Now we want to know the differences between the skeletal and cardiac muscle. They are shorter than the skeletal muscle, have just one nucleus, and also included the intercalated disc. The intercalated disc as these structures under microscope with hematoxylin and eosin. Some transverse line that located between of the cells. What is these structures? Intercalated discs are included some junctions that attach cells together and also communicate them together. For more communication between the cardiac muscle cells, the cardiac muscle, as in the first slide, have a lot of branching, cytoplasmic branching that connected with the adjacent cells as some intercalated disc. That's my max. 2 cells. Nucleus have a lot of cytoplasmic process. These processes interdigit together and form the intercalated disc. Why? Because the cardiac muscle cell should attach together very, very tightly. They don't allow to rupture or separate from together. Because the strength of the pulsing, of the beating of the heart, they should attach together very tightly by zonal adherent and desmosome. Do you know what is these junctions? Anchoring or adhesive junction. Yes, for attachment. They act as a glue. Yes, attach two cells together. Zonal structure is as a belt and the desmosome as a plug, as a bottom. But in the cardiac muscle, these zonula, these belts, a little are wider, have the more diameter and a little expanded, also named as fascia adherent, a little expanded for the attachment of the cells in more side, at least more power. And desmosome are some plug structures that attach cells together. These junctions are anchoring junctions for prevention of the pulling apart of the contraction activity of the heart. And other junctions that are in the heart are the communicating junction or gap junction. They communicate, connected cells together. Why? In the heart, guys, the beating, the pulse of the heart, yes, produce the heart by itself in the sinoatrial node. Okay? These impulses conduct to another node as atrioventricular node. And finally, some fibers spread out in all of the heart. he give to another students, and all of you finally give me. Yes, these impulses in the heart act as this conduct from one cell to another one by the gap junction that can spread very quickly, around 70 times. Yes, the heart beating in a minute. This conduction should be coordinated and more speed. For this coordination and communication, there are gap junction here that you can see schematically here. They are the zigzag light structures under microscope, electron microscope. They have some same as two states that this junction are in these intercalated discs. The intercalated discs is a specific structure in the cardiac muscle, not in other muscle. The SCR structures, the SCR are more numerous. The SCR have very fine tubules and not as developed as the skeletal muscle, but act as a storing of the calcium ion. The T-tubules are more in the cardiac muscle compared to the skeletal muscle. But the T-tubule and the cisterna of the SCR form a double structure, dyad. One SCR plus one T-tubules that located in the Z disc here. They included a high number of the mitochondria. Around 40% of the cytoplasm of the cells occupied by the mitochondria. And about the contractile protein, myosin, actin, and these proteins, all of these are same as to the skeletal muscle. Yes, they have more mitochondria. They have some lipid droplet and also glycogen that use this droplet or inclusion for the production, synthesis of the ATP, and also they included the lipofuscin pigment or aging pigment, because the cardiac muscle, as same as two neurons, are long-life cells. The waste of the metabolized process remain stay in the cells, and finally we can see these granules in the cardiac muscle. Under electron microscope, in the atrial muscle, these two are atrium, these two above cavity are atrium, these are ventricles. In the muscles of the atrium, the cells synthesis and act as endocrine cells. They synthesis one factor or hormone as atrial natriuretic factor. Atrial are in the atrium. Natriuretic means they involved in the increasing of the urine. When the blood pressure increased, or the volume of the blood or volume of the water in our body increased, these hormone synthesis and released by these cells to bloodstream that have receptors on the kidney and increase the existence of the natrium and following of it the water, and finally the blood pressure decrease. That is a one factor. Just this picture shows the electron microscope of the atrium. You can see these granules under electron microscope that are some dense granules that included these factors that act as endocrine cells. Endocrine means these products released directly to bloodstream. They don't have any duct and are just in the atrium, not in the ventricle. The last type of the muscle tissue are smooth muscles that occupy the wall of the internal organs, such as the stomach, intestine, the are flat. That's day by day contraction. They can move different things in the organs. Cells don't have any striation. It means they don't have any sarcomeric structures. Their cytoplasm included the actin and myosin. And the actin and myosin also have interaction together for contraction. But they are not arranged as dark and light line as sarcomeric structures. Because of it, they are smooth and don't have any sarcoma. The fibers are smaller than, yes, they are smaller compared to skeletal muscle and have mostly one nucleus. In the longitudinal section, with the flat nucleus, spindle-shaped nucleus located in the wider part and the narrower part are close to adjacent. In the transverse section, yes, when the knife passes from the cells, some part that pass from the nucleus, you can see round nucleus, and when the knife don't pass from the knife, some cells don't have any nucleus. They have one single nucleus and lots of mitochondria and other organelles that you now get. Then myosin and actin in the skeletal muscle and the cardiac muscle are in the sarcomere, yes? And the sarcomere have Z disc and M line, that the myosin and actin attached to Z disc, okay? But in the smooth muscle, they are not the Z disc. There are another specific structures in the smooth muscle that are dense body. That, as we see schematically, they are some dense structures that are in the cell membrane. The macromolecules of the cell membrane elytel are condensed and also included some types of the intermediate filaments as desmin or bimentin. Yes, in the cell membrane, the phospholipid proteins or other are more. They are dense and also additional to phospholipids and proteins, they included these two types of intermediate filaments. The desmin is a type of protein in all smooth muscle. In the digestive system, urinary system, reproductive system, muscular system, or others. But bimentin is just in the vascular smooth muscle, just are in the vessels, in the dense body. That finally, these intermediate filaments, desmin and bimentin, help to the actin and myosin filament to attach to the dense bodies in the smooth muscle. And finally, by the sliding of them, the whole of the cells shorter contract and whole of the muscle contract and shorter. Maybe this is I think is a combination. For example, one of the question may be the dense body, triad, and for example, the intercalated disc are in which of the muscle tissue, and we have, for example, you should memorize that they are combined together. For example, the right one is the smooth, the striated and cardiac. Three, don't know, maybe for example another one. There are which proteins are in the dense bodies, bimentin, actin, or the desmin, and for example the myomesin. Yes, these three are in the dense body. In the smooth endoplasmic reticulum, also the calcium ion stored in the endoplasmic reticulum, but most of the calcium ions that needed and necessary for contraction come from the calcium ion that are in the cell membrane. model finally resulted to contraction. It means in the smooth muscle there aren't any troponin, yes, because the troponin have a site for calcium, yes, but here the carmodulin have this role, yes, and they don't have any troponin. Finally, about the muscle regeneration, different type of the muscle regeneration, as I said for you in the first slide, some myoblasts remain as undifferentiated cells that we name them as satellite cells that are inactive and reserve myoblasts. These cells are few in the skeletal muscle. Because of it, the skeletal muscle are limited in their regeneration. It means when the injured site is very small, the regeneration occurs, but if the very big and severe, the regeneration not occurred in the skeletal muscle. Unfortunately, the cardiac muscle lack any satellite cells. Because of it, some person that suffered from the myocardial infarction or myocardial ischemia lost their heart function and need to eat some drugs or other things. A smooth muscle have lots of satellite cells and has good regenerative ability because they included lots of the satellite cells that can do mitosis and regenerate or repair the smooth muscle. The muscle tissue and the tissues, the type of the tissue finished.